come here. It's a great pleasure to give a lecture here. I will, in my lecture, give an introduction to the biophysics of hearing, and I will highlight the concept of critical nonlinear oscillators as amplifiers for nonlinear signal detection, which is a very general concept, but which is specifically relevant for the ear. So I will first give you an introduction to how the ear is organized and in the mechanosensory cells. And I will then sort of go into the discussion of the concept of nonlinear amplification. So the hearing organ sits in the cochlea in the case of mammals. And it is a sensory system that detects mechanical stimuli. So incoming sound waves elicit mechanical vibrations inside the cochlea, and then mechanosensory cells transduce these mechanical vibrations to electrical signals that are transmitted to the brain. And the signals that stimulate the ear are sound waves. And the remarkable feature of the ear and of hearing is the extraordinarily large dynamic range reflected in a broad range of possible amplitudes that the ear can process. So sound intensities, because of this broad dynamic range, are measured in a logarithmic scale because they cover many orders of magnitude. And so the measure of sound pressure level is therefore given in decibel. And the hearing range covers the range from 0 decibel to 120 decibels. And one can relate this to pressure amplitudes and to energy flow, sound intensity. So in terms of pressure, the sound pressure level is a logarithmic scale of the pressure amplitudes relative to a reference pressure, which is the sound pressure at the hearing threshold, which is about 20 times 10 to the minus 6 Pascal. And one can calculate the corresponding intensity in terms of watts per square meter transmitted by the sound wave, which is 10 to the minus 12 watts per square meter. And this corresponds then to 0 decibels. And the hearing range covers essentially 120 decibels, which implies 12 orders of magnitudes in sound intensity, from 10 to the minus 12 watt per meter square to 1 watt per meter square. And in terms of pressure, 6 orders of magnitudes. And the intensity is always a square of the amplitude. So in amplitude, we have 6 orders of magnitude. In intensity, we have 12 orders of magnitude. Now, this is a very remarkable ability of this sensory system. And it already hints at the fact that this must be a nonlinear system. Because if it were linear, like as a passive vibrator that somehow is set in motion and then read out by some nerve cells, 6 orders of magnitude in amplitude linearly would then transmit to 6 orders of magnitude in vibration amplitudes. And if you look at the possible ranges or the size of the organ, this could not work. So what I will discuss is nonlinear detection that is able to cover such ranges of intensity that are able to very strongly amplify the very weak signals while not amplifying the strong signals, and this over this huge dynamic range. Now this is achieved by, in mammals, by this organ called the cochlea. Has this interesting snail-like shape. It's a bone structure which has chambers inside, and it sort of winds up. This chamber is fluid-filled, and the organ that really detects the sound sits on this membrane-like structure inside these chambers. So if you take a cross-section and you blow it up, you see the so-called basilar membrane, which contains various supporting cells and sensory cells. And it also contains a sort of a material overlying it, elastic membrane-like material called tectorial membrane. So we have here the tectorial membrane. This is the basilar membrane with the sensory cells. And in the mammal, you have this characteristic structure that there are 4 rows of sensory cells, 1 row of so-called inner hair cells, and 3 rows of so-called outer hair cells, also schematically shown here. And you see already this sort of a linear arrangement. So if one unwinds the snail geometry, one can represent this as a linear structure, linear chamber. Of course, as a simplification of the real geometry. So the red line here is an indication of this basilar membrane structure. This is the fluid-filled chambers on both sides. And the arrangements of hair cells that you see here, they sort of run along this whole line from the base to the apex. And there are about 4,000 cells along each line. There are 4 lines of these hair cells, so there are about 16,000 hair cells in, let's say, human cochlea. Now, Van Bekkesi showed long ago that this basilar membrane can vibrate, and that vibrations sort of resonate at different positions along this linear structure depending on the stimulus frequency. So there's sort of a frequency gradient. The high frequencies resonate near the base, the low frequencies near the apex. So here an example, 100 hertz would be roughly here. The kilohertz, which is sort of the frequency where the ear is probably most sensitive, is somewhere in the middle, and the high frequencies are resonating towards the base. And there are sort of wave-like excitations or vibration of this basilar membrane that give rise then to these local resonances. Now, if we zoom in further, so look at these hair cells, these mechanosensory cells that you can see here. You can see these structures emerging from the surface of individual cells. So this is a single mechanosensory hair cell, and on its surface there is this set of hair-like appendages, so-called stereocilia, which are the organelles that are mechanosensitive. And you can see them here nicely as the characteristics of these individual cells. Now, in the case of the mammalian cochlea, they have this interesting V-shaped geometry. Here I show you an example of a frog, which is sort of more roundish. And so this is a scheme of these stereocilia, which are actin-filled protrusions of the cell, sort of rod-like objects that are bundled together in this structure. You see an electron microscope view of the whole cell. And this so-called hair bundle is a very sensitive mechanosensor. And it has ion channels that can be gated mechanically. And when they are open, ions can enter the cell and change the membrane potential, and therefore give rise to an electrical sort of signal. Now, this can be visualized here in an example from Jim Hudspeth, who uses here a dye that shows calcium levels. Here's a micropipette to sort of stimulate this hair bundle, and one can sort of here see visually the change in calcium concentrations due to opening of channels when the bundle is slightly moved. Now, the interesting structure that one can see between these stereocilia are these so-called tipplings, which are fine fibers connecting the stereocilia. And the current picture is that by shearing slightly the bundle, one exerts tensions on these tipplings and then sort of pulls open these ion channels. So there's a cartoon picture that is usually drawn, even though nobody has really seen in detail how this system is organized. So it may in fact be a bit different, but sort of the cartoon picture is sort of a sort of spring-like element pulling on a channel. And if it's set under tension, it pulls open the channel, and calcium and potassium can flow into the stereocilium and therefore into the cell. So first, to give you sort of a context of how the understanding of hearing evolved over time. It's of course a very classical subject of biophysics. And even though it's very old and many people have studied it for a long time, there are still many mysteries, and it's not really fully understood how this system actually works. So the original idea, sort of mainly due to Helmholtz, is that the cochlea contains sort of resonant elements, like, for example, strings of a piano, which then start to vibrate under sound waves, and that these local resonance vibrations are read out by some sensory cell. As I already mentioned, van Bekkesi then refined this picture in terms of collective wave-like excitations of the basilar membrane and slow traveling waves with position-dependent resonances. And since van Bekkesi did this work on dead cochlea, he missed the fact that the system cannot be viewed as a passive system, but is an active system that can amplify. And the passive properties are not really enough to understand the features of hearing, in particular this huge dynamic range, which I mentioned before. Now, since about the 80s, it is generally acknowledged that this system is an active amplifier. And this idea dates back much earlier to Thomas Gold, who proposed this already in 1948. But his idea was sort of ignored for a long time and then was revived several decades later when it was shown that he was right with his early suggestion. And the key discovery that sort of convinced people that the hearing organ is an active system and that the dead cochlea is not a model for how hearing works was the discovery of so-called otoacoustic emissions from living ears. And this is a feature that can be observed in many different vertebrate species, which all seem to possess active amplifiers in their hearing organs. And the characteristic is that if one puts a microphone into the ear canal of a human, of a mammal, or of some of the other species, one can typically detect emissions that are characteristic to the individual, that look very random. So here are some examples. So there are pronounced peaks at certain frequency in a sort of more or less random position in the spectrum, and they differ a lot from individual to individual. Now, the otoacoustic emissions are something which are only found in the intact live system. Similarly, if one looks at the mechanics of the basilar membrane, one finds that in the intact living system there are fundamental nonlinearities, which are sort of also associated with somehow active processes, because it requires not just passive material properties, and that are sort of key to this large dynamic range detection. And in order to see such nonlinearities in the mechanical properties of vibrations in the cochlea, sort of experiments are done by putting in a particular sound frequency, and then in different animal models where one can sort of open a hole in the cochlea and using different experimental techniques measuring tiny vibrations of the basilar membrane at a certain position in the cochlea. So there is a pressure amplitude with some frequency put in, and then the height, which is the variations or deformations of this basilar membrane, can be measured. And usually, for example, in sort of Mössbauer type techniques, it's a velocity which is measured. So the velocity is related to the height just by the frequency of vibration. And in such experiments, one sees very strong, clear signatures of a nonlinear response that hints at the nonlinear amplification. Let me just show you sort of an example of key data. So this is experimental data, sort of the membrane vibrations here in terms of velocity as a function of sound pressure level, decibel. And you can see that this experiment covers essentially the whole hearing range in sound pressure level, these 12 orders of magnitude of intensity. And it covers several orders of magnitudes in vibration amplitudes. Now, this is a sort of a log-log plot because we have a logarithmic measure of pressure and we have a logarithmic representation of amplitude. So slopes in this log-log plot correspond to power law type behaviors. And linear behavior corresponds to this blue line, and a sort of power law, which is a compressive nonlinearity, is less than linear, corresponds to this red line. Now, this experimental data is shown for the case where the stimulus frequency, which I think in this case is about 9 kilohertz or 10 kilohertz, you see it here, is chosen such that it's the sort of the best frequency at the place where the measurement is done. So this is sort of the resonant case. And in that case, one sees this pronounced nonlinear response. While if one does the same experiment at choosing a different frequency or with the same frequency at a different position, one would see a linear response over the whole range, which means this blue slope, or if you typically you have the same, for strong amplitudes, you get the same response in the linear case, and the linear response then corresponds essentially to this line here, which means for weak inputs, you get much less vibration amplitude in response as at this critical resonant condition. Now, you see here roughly a power law, which means the height vibrations scale like the pressure amplitude to the 1/3 power. And that is a very dramatic nonlinearity, which has deep consequences because it allows for the large dynamic range. Because as you can see, a power law of 1/3 would compress the 6 orders of magnitude in sound amplitude that correspond to the hearing range with 1/3 to just 2 orders of magnitude in vibration amplitude. Now, 2 orders of magnitude in vibration amplitude roughly corresponds to, let's say, from 1 nanometer to 100 nanometers, or from a bit less than a nanometer to less than 100 nanometer. But about this is sort of a range these hair bundles, you can imagine the hair bundles can perform. While larger ranges, they would not be able to perform without being damaged in the process. Of course, we know that at 120 decibels, actually hair bundles get damaged. So the 1/3 power law can achieve exactly compressing 6 orders of magnitude in only 2, which is what the system needs to be able to achieve that. And also you see this amplification by comparing to the linear response. So for the weak signals are amplified by many orders of magnitude, while the strong signals are sort of not amplified. So here you see this 1 nanometer corresponds to this, 100 nanometer corresponds to this, and we have the 12 orders of magnitude on the x-axis. Now, if you want to quantify sort of an amplification factor or a sensitivity of this detector, a nice measure is to take the ratio of output to input. We call that sensitivity. So that's in this case the velocity output divided by the pressure input. In a linear system, this ratio is constant for all stimuli. And that corresponds to the blue line. And from the power law response, if we divide h by p, p to 1/3 divided by p gives p to the -2/3. Yeah, that means that this amplification factor increases with the power law for weaker and weaker stimulus amplitudes. And that's exactly what you see in this experimental data. So this is sort of data which directly measures this, this sensitivity. One can see for very weak stimuli there's a linear response, which we also see in this data here. And then there's a crossover to this nonlinear behavior, which goes over many orders of magnitudes. And at high pressures, the system reaches the same behavior as the linear response when it is not at this resonant condition. So just to sort of summarize, we have several signatures of active processes in the ear that are all linked to the organ being intact and living. The compressive nonlinearity disappears when cells die. Autoacoustic emissions also. The nonlinearity then has a consequence that there are nonlinear combination tones. So if two frequencies stimulate the system, it will create additional frequencies by nonlinear couplings. That's, of course, a necessary consequence of having a nonlinear response. But, of course, both features together vanish with the nonlinearity in the system. And this, both of them depend on the intact living system. And also the sharp frequency selectivity that local regions possess depends on these active processes and is reduced in the passive case. Now, as I want to argue in the following is that these features can all be understood in terms of nonlinear dynamics as signatures of dynamic oscillators. Now, the simplest case of a dynamic oscillator is a mechanical system that can self-oscillate, that can produce spontaneous oscillations by its own. Of course, this requires an active system. The thermodynamic system cannot produce spontaneous self-sustained oscillations. And a system that produces self-sustained oscillations can usually be regulated by some control from the outside, and I usually use in the following a control parameter, which I call C, that somehow regulates the system. And in general, one can bring a system that can oscillate also to a non-oscillating state. And then one can draw such a diagram where the function of this regulation of the system, it can either be in this oscillating state where the resting state starts to become unstable and the system becomes self-oscillatory, and, and below this critical point, it is stable. And the green curve describes how the amplitude of oscillations continuously merges from zero amplitude at a critical point. Now, this is a behavior that one finds in many dynamical systems, a so-called Hopf bifurcation. And if one sits exactly at this critical point, such a system generally has very special and interesting nonlinear properties, and which essentially account for the observations that I just showed you. Just as a simple example of a nonlinear oscillator, just to help you think about how such a system might look like as a simple model, I show you here equations for a so-called van der Pol oscillator. A van der Pol oscillator is sort of based on the idea one can start from a harmonic oscillator with mass and spring and some friction. Of course, a harmonic oscillator is not a self-sustained oscillator. It will always damp to zero. It's a passive system. It can only transiently oscillate. But by sort of forcing the system with the feedback periodically at the right moment, one can effectively put this friction coefficient to drive it to negative values. And then one has an oscillator which becomes unstable. Of course, this requires an energy input. And if this were a linear equation like a harmonic oscillator, this would then be unstable and the amplitude would drive to infinity. But nonlinearities will stabilize such an instability at a finite amplitude. In this case, for example, nonlinearity would effectively introduce an amplitude-dependent friction and then stabilize oscillations at some steady state amplitude. And clearly, that such a system can be driven from a non-oscillating to an oscillating state by varying, in this case, for example, the parameter epsilon, which would be something like this control parameter on this axis. Now, this is not a model for the ear. This is just an example of a nonlinear oscillator that can have self-sustained oscillations. In general, many dynamical systems can have such properties. And I would like to discuss generic properties of such oscillators at the critical point very close to a Hopf bifurcation where the system becomes self-oscillatory. And just for the intuition, you can imagine if you have a system that is on the verge to oscillate spontaneously, but does not do so yet, if you stimulate it with an external force at the frequency that the system wants to oscillate spontaneously, you can do that very easily. There's sort of no response, no force opposing you doing that. And that's sort of this very high sensitivity that corresponds to stimulating a critical oscillator. Now, to explain these general features, let me do that on the blackboard. So I will now discuss general features of a Hopf bifurcation, in particular the response to external forces. So critical oscillators. And I will start with the response of a passive system to a time-dependent force. So I have a force f of t, and this then elicits a response x of t, a displacement. And I'm thinking of a very general system. It can be a non-equilibrium system, can be a passive system. For the beginning, all these are described by the same formalism. Now, the most general response function I can write down gives me x of t as a function of f. Now, for very small stimulus, there's always a linear response, which one can write in general in the following way: from minus infinity to the time t. I integrate over all past times. There's a response kernel t minus as a function of the time headed as past, and then the force at the earlier time. So all earlier times contribute to the current response. That's a linear response. But of course, if the stimulus gets stronger, the response becomes nonlinear. And this can be systematically captured in a Taylor expansion. So the next term I have to write as a double integral. And there are two time intervals. And now I have a response function which I call g2. This was a function g1. T minus t prime and t minus t double prime, f of t prime, f of t double prime. And this can be generalized to higher orders. Now, since we are interested in the following in periodic stimuli, I will use a Fourier sum for both force and displacement. So the force will be written as a discrete Fourier sum e to the I n omega t, and there's a coefficient fn in front. So these are the Fourier modes of the harmonics of a periodic stimulus. And the response will then also, after long times, move at the same frequencies with Fourier amplitudes xn. And in terms of these Fourier modes, the response function looks a little bit shorter. So I can write this response function by expressing the Fourier mode xn in terms of the Fourier modes of the force fn. The linear response is now just a complex number called g1 times fn. The quadratic term I have to sum over all frequency components k. I have a second order response function with two indices n k, and then f n minus k f k. So if one inserts these sums in this equation, the convolution here in Fourier space is just a product. And if I have a quadratic term, it turns out that only those frequency components that combined give the frequency of the nth harmonic, n omega, can enter. And therefore, so if you have e to the I omega, in omega, the mode n minus k and the mode k will together create the mode n. So the Fourier transform of this gives this term. And this can be also generalized to the next order, which is important, as you will see. We have to go to the third order to understand the system. Now we have to sum over two modes, KL. I have a third order response function, NKL. Now I have to sum over the modes that by free forces combined gives the mode Fn. I can write this as Fn minus k minus L, Fk, Fl, and then there are higher orders. So this is now a very general response function, which can be defined for any complex system that is stimulated and in the absence of force would not move. Now, we are now interested in the case where the system becomes unstable in terms of a Hopf bifurcation. And near the Hopf bifurcation, the linear response function sort of blows up, becomes bigger and bigger, because the sensitivity diverges. So we look at the case now where the mode n equals 1, which is stimulated with a force at frequency omega, has a sensitivity that diverges. In that limit, in that case, this Taylor expansion breaks down. It's no longer usable. So if you take a simple picture of x versus force, sorry, wrong diagram, x versus force. So the simple linear behavior would be like that. If now the response function diverges, this corresponds to the function becoming very steep near 0. And the Taylor expansion gets more and more problematic. However, if we just invert the relationship and just plot the diagram the other way around, the position here and the force here, then this steep curve becomes in fact a shallow curve. And this with a slope that goes to 0 rather than infinity. And in that case, so the inverted relationship is perfectly well defined as a Taylor expansion. And somehow it corresponds to inverting this relationship, writing the force as a function of x. You can also think of it changing the ensemble. Here we have an ensemble where we impose an external force as a function of time, and we obtain the resulting displacement. We can also think of the opposite experiment, imposing a time-dependent displacement and asking what is the force necessary to generate this displacement. And that's exactly the inverse of this function. It has the same mathematical form, but it has just x and f exchanged. So I call this linear coefficient f1. Now this goes proportional to xn. I have a quadratic term where I introduce the function f2. And now I have x n minus k, xk. Then I have a cubic term summing over two modes. Introduce the function f3 nkl xn minus k minus l xk xl, and higher orders. And the point is that in the vicinity where the G1 diverges, I can do this inversion and now have a Taylor expansion which doesn't have problems when I reach the critical point. Now, in principle, all the f's are related to all the g's, but in practice there's a complicated relation except for the linear response, where I simply have that f1 is 1 over g1. So as the linear response diverges, the linear response coefficient f goes to 0, and everything is regular here without any problems. Okay, so still, what I've achieved now, I have a very general nonlinear relation, input-output relation, that is applicable near an instability. And the instability has to do with the divergence of G1, or the fact that F1 becomes 0. What I'm now doing is I'm asking, what does the response between F and X actually look in the vicinity of this point? And this is completely general. It doesn't depend on the values of all these response coefficients. It depends only on the fact that they exist. So what I'm doing for simplicity now, I'm taking a sinusoidal stimulus. So I put only, so F1 is my external force amplitude at frequency omega, and all the higher Fs I put to 0. Now, in general, the system will now, because it's nonlinear, produce a response at the stimulus frequency omega, but also at all the higher harmonics. So I have, in general, xn. Now, if we go to very weak forces, the responses will be also small, because as x goes to 0, f goes to 0. And one can show, by studying this equation a bit, that if I look at the response xk, some kth higher harmonic of the response, this behaves or scales as the fundamental mode x1 to the power k. So if we have weak responses, higher harmonics are even smaller. So if x1 is a small number, xk is even much smaller. Because of that, we can make a limited expansion and just focus on the lowest contributions, the lowest modes. So I will therefore only, for simplicity, only consider the response of x1 and of x2, the first harmonic, which, as you'll see, is generated by quadratic terms. But the same argument can be continued to higher modes. So what I now do is, I'm taking this very general equation and only write down those terms that involve the modes 1 and 2, ignoring all the others. So then I don't have to go have infinite sums. I just have two equations. And then you'll see the basic features appearing. So I write the equation for n equals 1, and I write it for n equals 2. Only those terms that involve x1 and x2. Now, there's one subtle point I should mention. Because f of t is a real physical quantity, and the Fourier modes are complex numbers, there is a constraint that if you put a negative index n, where would I put that? If I look at x minus n, then this is the same as xn complex conjugate. In that sense, when I deal with x1 and x2, I implicitly also deal with x minus 1 and x minus 2. They are just the complex conjugates. Okay? But in fact, effectively, there are only two variables. Now, let's write first the equation for f1. That is the equation which balances the actually applied sinusoidal stimulus force to some system properties. So I write it down, f1 equals, now the linear term is there and cannot be avoided. And then there are only two quadratic terms that survive from this sum, because these are the only ones that create the fundamental frequency mode. 1, 2, and f2 1 minus 1. And they are proportional to x minus 1, x2. And you see the construction is such that the sum of these two indices must give n. So -1 plus 2 gives 1, which we have here. And these are the only two couplings which do that. And then there's a next order term is cubic. There we have also two terms which appear, F3 1 1 1 plus 2 F3 1 minus 1 1, and there the product is x1 square x minus 1, and note that x minus 1 is the complex conjugate of x1. Now these are all the terms that survive. This is exactly the number that survives. And we have a linear, quadratic, and a cubic term. And we have couplings of x1 and x2 in the quadratic term. Now the second equation for n equals 2 for the mode x2 starts with 0 force because I put the I use a sinusoidal stimulus here. There's no second harmonic in the stimulus. Then I have here the here's the 2, the second mode x2, and that's very easy. There's only one term remaining. All the others are higher order and can be neglected. There's a coupling term 2 1 x1 squared. So this is now two coupled equations for x0 and x1 for a given force. Valid in the limit of weak stimulus. Now what you can see here is sort of the stimulus force first excites, in particular we get this linear term, the mode at the same frequency x1. And the mode x1 that is generated via this quadratic coupling term in the second equation, that x1 square generates the second harmonic. That's the general feature. If you quadratic terms generate second harmonics, cubic terms generate third harmonics, and so on. So we can use this second equation to eliminate x2 from the first equation. So we can use this to understand how strong the mode x2 is, the first harmonics. And I just have to, I find now x2 equals minus f2 2 over f2 times x1 squared. And I can now plug this expression from x2 into x2 in the first equation. And then I have only a relationship between f1 and x1. So the dominant fundamental mode that is excited by the sinusoidal stimulus. What you see is when I plug x1 square for x2 is that the seemingly quadratic term in my expansion is of third order in x1. So it becomes effectively a cubic term. And I therefore lose the quadratic term if I only look at the dependence between f1 and x1. So what I get then is a relationship f1 equals some linear coefficient which I call a in the following times x1, and the first nonlinear contribution is cubic. I call the coefficient b. Now the cubic term always has the form x1 squared times x minus 1. These two become the same. And x1 times x minus 1 is a modulus squared. So I can write this as modulus x1 squared times x1. And by just comparing these two equations, I find expressions for both a and b in terms of all these linear and nonlinear response coefficients. So a is simply given by f11. And I should like to mention that this is a function of frequency. This is a linear response function, which in general depends on frequency. And of course, in real time, this then corresponds to the fact that the linear response function has a memory kernel. Now, b is a little bit more complicated, but let me just write it down for you because it's so general and beautiful. So b I simply get by taking all these cubic nonlinear terms into account. And it looks like F211 over F21 times F122 plus F1 minus 1, 2 plus F3 111 plus 2 F3 1 minus 1 1. So while A is very simple, this coefficient in front of the nonlinear term is a quite complicated combination of quadratic and cubic nonlinearities in the system, and also linear response coefficients. And but interestingly enough, even though the system in general has quadratic terms, our genetic response between the force amplitude and the response amplitude has no quadratic nonlinearity. The cubic nonlinearity is the first one. Now, this already hints at these nonlinearities and power laws that we find in the ear, because if we go to the point where the system is a critical point, I mentioned already that the linear response coefficient F vanishes. Therefore, the linear response goes away, but only at that, if stimulated at the right frequency. And then the relationship between F and X has a 1/3 power law. So F proportional to X cubed, or X proportional to F to the power 1/3. Now, in practice, of course, we also may drift off a little bit from the bifurcation, and the A is there. Then we have a more complicated relationship, which for small amplitudes is dominated by the linear term, because X is very small, X cubed is even smaller. While for sufficiently large amplitudes, the response is dominated by the nonlinear term. And that's exactly what we see in the experimental data. Now, this is a simulation of such a model. The data, you see here the linear behavior, and then you cross over to the nonlinear behavior. So let me also just clarify 2 points a bit more. Now, this equation contains all information about the oscillator near the bifurcation, and it also contains the fact that it actually starts to oscillate beyond the bifurcation. Now, let me briefly point it out to you, and I'd like to also show you that this is equivalent to what is called a normal form of a dynamic system near the bifurcation. So in nonlinear dynamics books, you will find derivations of a normal form of the dynamics near a limit cycle, Hopf bifurcation, where a limit cycle appears. This calculation is sort of equivalent to these alternative approaches that are usually used in nonlinear dynamics. But this approach here, which I show you, also gives you the coupling to external forces, which in a standard approach is not so easy to get. So I already mentioned that if we go to the critical point, then A is 0. And if we expand near the critical point, then A of omega and the control parameter C can, to linear order, be written as some coefficient which is complex alpha times omega minus omega C plus some coefficient beta times C minus C, C. So that's true linearized near the critical point. At the critical point, both the control parameter and the stimulus frequency are matched to their critical values. Then A as a complex number is 0. And then we have a response that the modulus of x1 is b to the minus 1/3 times f to the 1/3. And that's exactly this cube power law. Another observation is that this equation can also have spontaneously oscillating solutions. And in the absence of an external force, spontaneously oscillating. So if you take f equals 0, we can divide the equation by x1. and we find that there may be solutions with an amplitude x1, which is spontaneous because there is no force, which has a magnitude minus a over b. Now, at the critical point, a is 0, and there is no spontaneous oscillation. Now, in order to get a spontaneous oscillation, we must have that a over b is real and negative because of the minus sign, because this is forcibly real and positive. Now, this then limits the values of c and, or in fact, this then determines the value of omega. So this depends on omega, and only for certain frequencies omega will this ratio become a real number. And if we can plot as a function of the control parameter, we then get sort of a line of the frequency of the system that allows for this. Now, along this line, a over b is real, but only along a certain part of the line, we actually get a positive number. And only there we have actual oscillations. And if you sort of draw the diagram of control parameter versus oscillation amplitude, this is the critical point where the oscillation starts. So this provides the frequency of oscillation as a function of control parameter, including the finite critical frequency at the critical point, the critical point itself, and it describes the response of the system to an external stimulus near or at the bifurcation. Yes? So the control parameter is any parameter that you choose that if you vary, the system changes its properties. And since, so as I'll show you in a moment, this system has a lot in common with phase transitions. And usually if you do classical phase transitions, you vary the temperature. And then at a certain temperature, you get a phase transition. Now here we are not in a thermodynamic system. It's a non-equilibrium dynamic system, which also has a phase transition. And you vary, in general, other parameters to go across the transition line. And what I'm describing is totally general. So here I do not want to specify what the control parameter is. If we go to a biological system, we have to find out what are the parameters that, for example, a cell actually changes and controls in order to control the dynamics. And in the hair cell, one suggestion would be a calcium concentration. But that would be a specific model. Yeah.
Essentially, you have shown that the quadratic term disappears.
Yes.
It looks to be completely general.
Yes.
I mean,
that's the beauty.
Any kind
yes.
Of, okay.
It's true for any system
and
that
even if you put
that
more,
can oscillate.
More terms.
Yes.
Okay.
Yes. It's totally general, but of course, what I'm doing here is I expand around a point where the linear response vanishes for one frequency, and that is then automatically a Hopf bifurcation. Yep. Other questions? How am I doing with time? Okay. Yeah, I could also briefly mention that from this relationship follow a few other interesting facts. In practice, of course, A is not exactly 0, either because the stimulus frequency is not exactly the critical frequency, or because maybe the system is not perfectly tuned to a critical point. As I hope I can show you also later, in the presence of noise, A may also not be 0 completely, and so fluctuations bring the system slightly off this ideal critical point. Now, if A is slightly non-zero, for small forces, the system has linear response. For large forces, it has nonlinear response. And from this relation, we can also see that there is a characteristic crossover force, which we see in the experiments. And we also find that if we take a frequency mismatch, there's a range of nonlinear amplification that depends on the amplitude. So if we have a big frequency mismatch, we need larger forces to get to the nonlinear regime. And there is a force-dependent frequency range in which the system amplifies non-linearly. And the bigger the force, the broader the frequency range where amplification is nonlinear. And if the force gets weaker and weaker, the nonlinear amplifying regime gets narrower and narrower. And these features all are generic and included in this description. I maybe use also a moment to show you also that this form is completely equivalent to this normal form of a Hopf bifurcation that is often used. Let me just erase this part here. So what I'm going to do is I will use this relation and I will use this linearization of A. So which means it only works close to the critical behavior in terms of frequency and control parameter. And it is useful, alpha is a complex coefficient, it is useful to pull out the factor I here, the imaginary factor I, as you will see in a moment. So I will write A as a phase, which I call theta, and a modulus of alpha. And then I write it as I omega minus I omega c. And so I've chosen to represent alpha by pulling out an I, its modulus, and some phase. And I leave beta as a complex number times T c minus cc. Writing A in this form, I can re-express this equation, the basic one, as I omega x1 equals I omega minus I omega c plus R x1 plus, I call this little b, I think there's a minus here, x1 squared x1. I'll explain in a moment what I write, e to I theta divided by modulus of alpha f1. So I've only re-expressed the equation. And I have divided the equation by this prefactor in front of I omega. Therefore, I have redefined B, and I have this prefactor in front of the force. So it's the same equation, but now if we introduce a complex variable z, which is time dependent, write it as x1 e to the I omega t, so that's our mode amplitude, then we can interpret this as a dynamic equation for z. So the I omega x1, I can interpret then here as a dz dt term equals I omega c plus r z minus b z square z plus e to the I theta over modulus alpha f of t. And that is what is called the normal form near Hopf bifurcation. And R is related to this term here. I don't want to write down the exact expression. Maybe I do write it down. R equals the real part of beta over minus I alpha times C minus C critical. So this term is related to being off from the critical point by the control parameter. The little b is related to the capital B by the same factor that you find in front of the force. And you see also that the force enters the equation by some phase factor, some phase shift, which is a property of the system and has to do with the complex nature of the linear response coefficient and this coefficient alpha. Okay. So that sort of completes the picture, because that's a version that you will always often find in books about how to describe Hopf bifurcations. Now, so I showed you that this concept explains the types of behaviors that are seen in the ear, and here reproduced by a model. So linear response, if you're off the right frequency in the stimulus or off the critical point in the control parameter, and as you approach the critical point, this linear response moves to give way to this nonlinear response, which is compressive but much higher, and you get this amplification. Now, one important issue in this context of biological system is to ask, why are these oscillators critical? So if the system operates somewhere away from the Hopf bifurcation, this is not good for detection. If it's on the non-oscillating side, it will lose some of the nonlinear response. You will see more of the linear response, in particular for weak stimuli, and you will lose their detection. If you're on the oscillating side, then the system itself produces vibrations, even in the absence of a stimulus, and this then interferes with the external stimulus. It's difficult to interpret for the brain. So best is if the system operates exactly at the critical point, and for that, this frequency has to be, the control parameter, whatever it is, has to be regulated to the right value. And I just want to very briefly mention that one can think of self-control mechanisms, feedback loops, which keep the system at the critical point. And this is sort of illustrated here. So we have a system that has an oscillatory or non-oscillatory behavior as a function of some control parameter. And I mentioned before, in a cell, this control parameter might be a calcium concentration, which changes when ion channels open in the hair bundle. Now, calcium is also pumped out to maintain things at a good level. And one can use that to regulate the system to close to the critical point. So I would then introduce an additional dynamics for the control parameter, which should be slow. And you can imagine that as long as the system is in the oscillating regime, sort of you vibrate spontaneously, and therefore your calcium enters. This will then increase the control parameter. So this is an open probability of some channel, and you will drift away from the oscillating regions towards the non-oscillating regions. After you pass this critical point, the system is in the stable state where it doesn't oscillate, and therefore there's no longer calcium coming in. Sort of the term outpumping, which decreases C, takes over, and the system drifts back towards the oscillating regime. And there's a steady state very close to the critical point, slightly inside the oscillating region, which is reached by such a self-regulating feedback. This system also has some interesting properties which I think correlate well with physiological observations that are often called adaptation or fatigue. So if you are subject to loud sounds and then you switch quickly to a very quiet environment, you will be less sensitive to the very low intensities. And this could, for example, correspond to being sort of in a loud environment. This system would detune away from the critical point. And then it will take some time in a quiet environment to move back again to the critical point. So in that sense, this feedback would contribute to being able to detect very faint sounds only if you had a moment in a quiet environment to adjust the system to criticality.
So
yes?
What is underlying such a discrete feedback?
Oh, this is just an example. In practice, it's not discrete, but it's easy to discuss with a very sharp step here. But of course, in practice, it will be a smooth curve. Yeah.
So it's not known about precise mechanism?
No. I'm speculating here, and I'm just mentioning such systems might exist, but we have not yet been able to point our fingers to them actually existing. Except that there are signatures of such systems in the phenomenology. Okay, so far I have, yeah.
So if I want to draw an analogy between this critical point and a thermal phase transition, so what plays the role of diverging fluctuations in the thermal phase
I'll come to that later in my slides, so maybe it's better to postpone this for a moment. I hope the time doesn't run too fast, but I'm sure I will not forget this. Let me first talk a little bit about hair cells, because one of the first questions that comes up in this context is, what is the oscillator? What is the dynamic system that can undergo Hopf bifurcation in the ear? And probably there are many of them coupled together. It's a very complex system in the end, as I'll also show in my talk. And the first thing I'd like to mention is that the hair bundle itself of a hair cell can be a mechanically active system and can actually oscillate. And this was discovered some time ago by Pascal Martin and Jim Hatzbeth by studying frog hair bundles. And I just show you here one example from Pascal, where you see under a microscope 2 hair bundles, light microscope, so they're not very well resolved. This would be an electron microscope view of these structures. And you see them stochastically vibrating. Here you see a time trace, and you see a spectrum. And this, already on the level of a single cell, one can now study aspects of the type of physics that I've introduced. So what Pascal has done is to stimulate these hair bundles with an elastic thin glass fiber. So sort of imposing an external force, which you can calibrate by knowing the stiffness of his fiber. And then he can impose a periodic sinusoidal force onto the structure, and then look at the response. And so the force is now just a single Fourier mode in the sense I described on the blackboard. And then in order to get the response x1, the mode that is excited, this is just the Fourier amplitude at the stimulus frequency, which one can pick out from that such signal. Of course, there are also higher harmonics. And one can measure the sensitivity, one can measure the response. So all the quantities I discussed, one can measure here. And one can see the signatures of this nonlinear behavior. In particular, one can see if the system is stimulated at the frequency at which this hair bundle vibrates. In this case, a very low frequency, about 8 hertz. But these low frequencies hair bundles are very good for experimental study. But also, they oscillate very nicely. One can see that the sensitivity goes up dramatically. So that's a nonlinear response. While if it's stimulated at different frequencies, one has a linear response, which is much weaker. Now, this is a single hair bundle. And one can also plot this sort of in the log-log plot. One can sort of see hints of this power law behavior, but it turns out to be quite small amplitude range where this works. And this has to do with there is a saturation region up here where one loses the nonlinear behavior, and the linear behavior sort of cuts off this power law at small forces. And so I have about an order of magnitude of this power law, at least as a signature here. But clearly this is we call this gain, the difference in amplification factor at high and at low stimuli is in this case about 10. Now let me maybe briefly just mention, don't want to go into details here, that one can understand many aspects of the hair bundle dynamics as a dynamic system which has nonlinearities due to the nonlinear mechanics of channel gating, ion channel gating, and it has an energy source and an active behavior because of myosin motors that move in actin filaments and that can also generate forces on the tip link and serve as an active mechanical element. And then there is a feedback in the system, which is the regulation of myosin motors by calcium. And calcium itself flows into the channels when the system is stimulated. So then this gives us sort of a dynamic system with two coupled equations that can have an oscillating regime. So this is a state diagram with an oscillating regime, closed and open channels, bistable state. And here it's really oscillating. And along this black line would be a Hopf bifurcation. But in order to understand the experiments quantitatively, one has to take into account fluctuations, which I have ignored in all my discussion here on the blackboard. And there are different sources of fluctuations. There's motor noise, there's thermal noise. We can estimate the noise strengths in the system from physical arguments. And having a noisy system essentially amounts to adding in this dynamic equation here a noise term. You can add here a noise term with sort of noise correlations. And this noise introduces the fact that this bifurcation that exists in the noiseless case is sort of concealed to some sense because the spectrum of the system now is not a delta p at the oscillation frequency, but it gets broadened. And the width of the power spectrum of spontaneous movements has to do with the correlation time over which phase coherence gets lost. And this is also a relaxation time that can be read off from the width of the spectrum. And since the system at a Hopf bifurcation, which is a real critical point, becomes slow in the sense that relaxation times go to infinity, any oscillator with a finite width spectrum will not go to this ideal critical limit. And there is a finite relaxation time, which corresponds, in fact, effectively to a coefficient R in this equation, which is similar to having a mismatch from criticality. So in that sense, the system cannot be exactly critical with noise. And that's one of the reasons why we never can get rid of this linear regime for very small amplitudes. However, this model can account for the statistics of the observed stochastic movement. So this would be an experimental time trace. This is this model. And this would be the spectrum of the model and the spectrum of the experiments. And it can also account for the linear response functions. So I mentioned that the linear response function, which is called A, is a function of frequency, is a complex number. One can write both the real and the imaginary parts of this linear response function. In this case, it's a function of frequency. And one can measure them, these are the data points, and we can calculate them in this model. And this works extremely well, but the noise is essential. It would not match without noise. So this is experiment, this is simulation. And here you see also the nonlinear response in experiment and simulation. So we have a model for a single hair bundle that can account for the observed phenomenology. But because of the significance of noise on the single scale level, exhibits the features, but with a much reduced gain. So in the cochlea, we see a factor of 1,000 between the highest and lowest amplification. In the single hair bundle, we see only a factor of 10. And that's an important open question that I can talk a little bit about in what comes. But now I come to the question that was raised earlier. So what is the relationship between classical phase transition and the critical point I've been discussing so far? So classical phase transition is best sort of discussed in the context of the standard toy model, the Ising model, where we have a thermodynamic system with spins on a lattice. There's a neighbor interaction, and on very large scales, there's universal sort of robust behavior that doesn't depend on molecular details. In particular, there is a critical point at a particular temperature at which a correlation length diverges. Spins get correlated over large distances, and spins and their fluctuations. And there's also a relationship between the magnetization and the magnetic field, which is a power law. And one can distinguish so-called mean field approximations from sort of full treatment of this problem. And in the full treatment, one gets slightly different exponents or different exponents as compared to mean field arguments. Now, this is an equilibrium system. Now, the point one can make is there's a complete analogy to our oscillator system, which also reflects at the Hopf bifurcation a phase transition, but a non-equilibrium phase transition. So we can think of a lattice of many noisy oscillators like our individual hair bundle. Each of them, as a single system, fluctuates a lot and doesn't exhibit a real singularity as one needs for a phase transition. But if I couple many of them together, I can get a spatially extended oscillating medium in which, as a function of some control parameter, which in non-equilibrium case is no longer temperature, It's usually something else, and depends on the particular system of study. That as a function of the control parameter, the correlation length, the length over which phases are correlated in the system, diverges, and when the correlation length diverges, these oscillators start to synchronize. So over the correlation length, they are in synchrony. Beyond the correlation length, they are not synchronized. And there is a critical exponent for this. And similar to the relationship between magnetic field and magnetization, we now also have a relationship between vibration amplitude and forcing. So the magnetic field corresponds to the force. And there's again this non-trivial exponent. And what I discussed on that blackboard then corresponds to the mean field theory with delta equals 3, 1/3 exponent. And of course, one can generalize this idea and go beyond mean field theory of really a spatially extended medium. And doing that, one actually finds that a system has the same exponents as a thermodynamic system, but some additional features which clearly show that it's out of equilibrium. And the phase transition only exists, as in thermodynamic systems, in the limit of large system size. The number of oscillators has to go to infinity. In that limit, one can get a true phase transition between a state that loses phase coherence on a finite time, oscillators are not really synchronized, and a state where all the oscillators have a common frequency and a sharp delta function in the spectrum. So if we are in the stable state, we have correlation functions that decay, corresponding to a loss of phase coherence over a certain correlation time. And in the spectrum, we have then sort of broad peaks, like you saw for a single noisy oscillator, which loses phase coherence over time. And if we now go to the critical point, suddenly from this broad spectrum emerge 2 delta peaks, which is really a coherent oscillation. And these delta peaks describe sort of the coherent oscillating phase, the mode that the system collectively generates at well-defined frequency. And at the critical point, we get power law spectra. Yeah. Now, one could use this type of physics to improve the quality of these oscillators that we see single hair cells are, to get a better working nonlinear amplifier as a collective system of several oscillators. And there is a reason to do that, because in the ear, hair cells cannot vibrate independently. They are actually coupled, in particular via this overlying tectorial membrane, which is an elastic structure. So the simplest picture would be to ask, how does a group of hair cells behave when they are coupled elastically? And so one can use this model of hair bundles that I've briefly introduced, and then add springs between them, sort of corresponding to the tectorial membrane. And this is sort of a movie from Kai Dirkes, who did the simulations. So these are independently stochastically oscillating hair bundles, which are now coupled by springs. And so what happens when one couples these noisy oscillators with neighbors? Yes?
What were the interactions in this model, the oscillators on the lattice? What kind of interactions were there? Was it like
Springs. So they're just elastic springs attached between them.
Yeah, in this example, but I mean in this model a few slides before. I mean, do they synchronize their phase or their?
Here you would also, in a general model, you put a linear coupling term with a complex coefficient, which describes both elastic and viscous couplings. And it's a very general equation. It's a complex Ginzburg-Landau equation with noise, which describes the generic features of such systems. Yeah. But here's now a specific model for a hair bundle. Now, if you just look at the spectra of spontaneous oscillations, so this would be a without coupling. And here we change the coupling strength between cells, and then the spectra change, and they get more sharply peaked, which corresponds to a situation where this phase coherence time gets longer, and the system looks more like a critical system. So that suggests that these systems which oscillate when these hair bundles are coupled will become a better nonlinear amplifier, and that is indeed the case. So here we see how this sharpness of the spectra depends on coupling strength, for different groups of coupled hair bundles. So here these are 4 hair bundles, these are 9 hair bundles. This will be the single one. And this shows then the sensitivity as a function of stimulus for different groups of coupled hair bundles, showing that one can increase quite significantly this amplification gain, but one needs also significant groups of hair bundles working together. So for example, in the case of 9 times 9, when one gets values similar, approximating almost the types of values we see in the ear. But one should also not compare these two models, because in the ear we have a much more integrated complex system. And also the hair bundle model that we used was really developed with the parameter choices for bullfrog hair bundles. We do not understand well enough mammalian hair bundles. So it's difficult to speculate how the mammalian cochlea really achieves this huge gain, what the difference actually is. So we think that coupling of hair bundles with this tectorial membrane plays a key role. And in our study, we, for example, looked at systems where sort of the outer hair cells, 3 rows of them along about 20 cells are coupled together, and this can produce sort of effective amplifiers that have very good properties of amplification, coming close to what we see in the ear. One should also add that, I don't have time to go into this, that mammalian outer hair cells, these are these 3 rows of cells which presumably do this amplification in the ear, have another very important and interesting feature, which is called electromotility. And electromotility means that they are piezoelectric elements, that if you change the membrane potential, can contract their whole cell body. Now, this is essentially a linear process, but it has dramatic consequences for the system, because it much more strongly couples hair bundle movements with the feedback to basal membrane vibrations. So if the hair bundle moves very slightly, this generates changes in the membrane potential, and this then generates the mechanic contractions of the cell and then vibrates the whole basal membrane. So this provides an additional feedback in the system, which changes the properties of the unit elements. And this is also not very well understood, how this can contribute to this noise reduction that we need to get the high amplification at very weak stimuli. So this, I think there's still a lot of work has to be done to understand the interplay of the properties of hair bundles and electrical motility, and to understand really the source of the nonlinearity. So far, our idea is that the main source of the nonlinearity comes from the ion channel nonlinearity, when they open and close. And electromotility is an almost linear piezoelectric phenomenon, which cannot really contribute nonlinearities. But all of this is still not resolved. Now, in the remaining minutes, I'd like to sort of complete the picture of the mammalian cochlea by taking seriously the fact that there are many oscillators that are coupled together. So far, I have discussed the problem by just using a single critical oscillator as an example for a system that can explain what happens at the resonant place. But in practice, the system is much more complex and integrated, in the sense that along the whole cochlea are these detectors and are these active amplifiers. So we need a set of critical oscillators, critical dynamic oscillators, distributed along the cochlea with a gradient of intrinsic oscillation frequencies to realize the system. And of course, that's a much more complex system than a single oscillator. They're all coupled, and there are waves that are propagating. So this can be done quite elegantly by using a linear model of the cochlea, ignoring the three-dimensional geometry and also the spiral. But it's very instructive because it highlights the essence of the problem. And sort of motivated by earlier work by in particular Swislocki, Lighthill, Zweig, and De Boer, with Tom Duke, we used a one-dimensional model of wave propagation, taking into account critical oscillators. The idea is we have a fluid-filled chamber there are two pressure profiles, P1 and P2, in the two chambers. If the membrane vibrates, fluid has to move, and the volume flow of the fluid in these chambers is called J1 and J2. Now, what counts in the end, to write down the dynamic equations for the wave, is only the difference in pressure across the partition and the difference in the volume flows. I call this capital lowercase p and lowercase j. And maybe I motivate one step on a blackboard, because it's quite beautiful, the basic physics of this cochlea. So to understand the basic physics, there are two relationships. There's a mass density of the fluid in the chamber. And if there is a fluid flow, I have an inertia term, which is proportional to the mass density and the rate of change of the fluid volume flow. And this inertia term that acts in the fluid is balanced by pressure gradients. And if I take into account the geometry of the cross-section, I have the cross-section area, and I have a position dependence of the pressure. So pressure gradients are balanced with the inertial forces. This is a little p, and this is a little j. So I can do this for each chamber separately, and if I take the difference, I get this equation. In addition, there is a conservation law, because the fluid is incompressible. So whenever the membrane is displaced, fluid has to move elsewhere.
Is it true that the cross-sectional area is constant?
It's not constant, but these are then subtleties which I don't want to go into. Yeah, it's actually position dependent, which makes the equations a bit more difficult to solve, yes. So the second equation is that the width of the channel B times the rate of change of the height, that's something like volume that is displaced locally by height change. And this then has to be balanced by a flow. So there has to be a flow, otherwise it's not possible because the fluid is incompressible. Plus dxj equals 0. And if you do it for the two chambers, you get a factor of 2 here. So we have a force balance in the fluid, and we have a conservation law. And if we now take the second derivative of h, so the first time derivative of this equation, and the first space derivative of this equation, then we get these cross terms are the same, dx dt and dx dt here. So I can then eliminate the volume flow. And I get an equation 2b rho dth squared, the second derivative of the height, is equals to bl, the second positional derivative of the pressure. Yeah. And this looks almost like a wave equation. That's why there are waves in the cochlea. So I have that here, this equation. And if now the cochlea were a passive system, where the local pressure deforms the basilar membrane, so the pressure difference across the partition deforms the basilar membrane, and K is a mechanical elasticity of the basilar membrane, then we can plug in this expression for P, and we get a wave equation for H. And because this stiffness depends on position, the wave propagation velocity depends on position. If one adds a mass term here in the passive case, that was sort of the classical picture, you can get local resonances. And it's a quite rich wave physics that one can find in the system. Now, what we have to do to understand nonlinear active behavior, so that would be now the Fourier space. I look how the system responds to a single frequency omega in frequency domain. And in that case, it's very easy to introduce my critical oscillators, because I just throw away the passive relationship between pressure and deformation and replace it by a critical oscillator. So what was the force before is now the pressure difference across the basilar membrane. What was the x before is now the height change of the deformation of the basilar membrane. I have a linear term and nonlinear term. And I can now plug the p in here. And what I get is a quite beautiful nonlinear wave equation. This is now an equation for the propagation of nonlinear waves. This equation is a difficult equation to solve. And because it has position-dependent coefficients, here for simplicity I ignored the position dependence of the geometry, and it has a nonlinearity, and it has also the possibility to spontaneously oscillate in certain regimes. If you choose parameters inconveniently, this thing can self-oscillate and emit waves. It's sort of it can become the acoustic analog of a laser because of its active nature. Now here you see a few examples of numerical solutions to this equation. So these are now wave-like modes. And because the system is nonlinear, the wave patterns have a very different shape at different amplitudes. So this corresponds to 40 decibel sound pressure level, this corresponds to 80 decibel sound pressure level. And here are 3 examples at different frequencies. So they resonate at different places. If the amplitude goes up, the range of resonance becomes wider, and that's what I described before. For larger forces, the frequency mismatch that where you still amplify non-linearly gets broader, and therefore you change then the shape and you get less sharp resonances. And this model can account for many of the basic features that are observed in the cochlea. So the key properties of the traveling waves and the nonlinear response and the dynamic range. So here I just compare the displacement amplitude as a function of frequency in a model, and here some data measured in the cochlea. Now many details are hard to capture by such a simple one-dimensional model because the three-dimensional nature is relevant to the problem. Maybe just some final comments. So what one has to do to define the linear response, it depends not only on frequency but also on position. And that is because the oscillation frequency must have such a frequency profile, because on one side of the cochlear system is sensitive to high frequencies, on the other side it's sensitive to low frequencies. The frequency profile is to a very good approximation exponential. There's a characteristic distance d between one octave. And if I use my linear response function with the coefficient alpha and I linearize it, I now have a resonance frequency which depends on x. This is my stimulus frequency, so I get a quite complicated function A of omega on x. And in the general case, I can also sort of sketch how such a linear response function must look like. Because if I take the real and the imaginary part, which I call A prime and A double prime, of the complex function A, the real part, A prime, must be symmetric in terms of frequency, and the imaginary part must be antisymmetric. Furthermore, because the system is a critical oscillator, at the characteristic frequency at the position x, both real and imaginary part must vanish. Therefore, we know what the qualitative shape of this function A must be. Even though we don't know exactly what it has never really been measured. But it must go through 0 here. One is symmetric, the one is antisymmetric. And what one sees now is that if we have a wave entering the cochlea, it will propagate along the basilar membrane. And if it's far away from the resonant point, it's outside of the nonlinear regime, and it's a linear wave. Now, for this linear wave, this linear response function is key for the propagation. And since the imaginary part for frequencies smaller than the local resonance frequency, which is the case when here are high frequency cells and a low frequency wave propagates by, and its resonance is further down the lower frequency. So if the frequency is lower of the stimulus than the local characteristic frequency, we have a negative imaginary part, which corresponds to negative friction. And this negative friction actually pumps the wave. So we have a wave propagating through an active medium, a linear wave at this point. The nonlinear dynamic oscillators pump the wave, put energy into the wave because of this negative friction coefficient. Then the wave propagates until it reaches the critical frequency where, because this goes to 0, the nonlinearities take over and then set the amount of vibration. So automatically, before the resonance point where the nonlinearity happens, there is an active region where the wave is pumped. This, of course, can help also increase the magnitude of weak amplitude waves, or also overcome effects of viscosity in the fluid chamber, and also viscosity in the basilar membrane. Yeah, I think I'm over time. Yeah, so let's have 1 minute or so. Just to mention, to finish, to come back to the autoacoustic emissions introduced earlier, it's still an open question of what is the origin of autoacoustic emissions. And there is a debate of possible mechanisms and origins. I'd just like to finish in showing a recent work where we've shown that we can account for the autoacoustic emissions in the context of the model that I just introduced, with the idea that there is disorder in the cochlea, in the sense that the control parameter that tunes oscillators to criticality may vary in a disordered way. So we have a static random deviation from zero as a function of position. So there are regions in space where oscillators tend to oscillate spontaneously, while there are other regions in space where they are more stable. This is sort of, in each individual cochlea, the stochasticity is different. It's sort of a disorder in the system. But as a consequence of having regions where system is locally not critical but towards oscillating side, this gives rise to clusters of cells because of the coupling that I described before that oscillate in synchrony. And between these groups which oscillate in synchrony, there are jumps and then plateaus for other frequencies. And each of these plateaus then corresponds to a peak in the spectrum of spontaneous noisy activity of such a cochlea. And one can recapitulate the statistics of autoacoustic emissions in humans with this model. Such as, for example, the statistics of typical intervals between autoacoustic emissions, which is now set by the coupling strength of elastic coupling between hair cells. And I don't have time to go into this, and I'd like to finish with this and with acknowledgements to the people who have contributed in many years to all these projects and works. So thank you very much.